All right, so this is your membrane transport notes part two, and we're going to focus on this back side and talk about transport today. So we're going to start on the left here with this third, and then we'll move towards the right side as we go on. So cells have to move materials both in and out, right? If the cell needs material that is mostly outside the cell, it has to come in, and if it's mostly waste products perhaps that it has to get back out, it has to move across the membrane also. And ideally, cells are going to spend the least amount of energy to do this, right? Energy is expensive. For us, we might have kind of like an endless supply of food available, but evolutionarily, energy is not something you want to waste. So what you're going to see is a lot of these processes are going to minimize the amount of energy that they need to occur. So we're going to start here on the left with passive transport. Passive transport requires no energy. So we can go ahead and put that. No energy required, and by energy we mean ATP. No ATP required, right? So if things are diffusing, this is just the natural motion of molecules and kinetic energy, right? So basically, if you have high kinetic energy, or sorry, if you have a high concentration of molecules, you have all these molecules all adjacent to each other, and they're all moving around because they all have heat and kinetic energy. They're going to bump into each other, and the end result of that is for them to disperse. This is like if somebody sprays perfume in a room. Right out of the bottle, it's super high concentrated, but it diffuses across the room because of those kinetic energy and the collision between the molecules, right? This is a completely natural process. It requires no ATP, and it happens spontaneously. So with passive diffusion in cells, when is this going to happen? Right, when is this going to happen? This is going to happen when you have non-polar molecules and hydrophobic molecules. And the reason they have to be non-polar hydrophobic is because these molecules are going to move through the membrane without any help, simply just based on kinetic energy. And to do that, they have to make it through the hydrophobic fatty acid tails in the center of this membrane, which means they also, whatever these molecules diffusing are, they must also be non-polar and hydrophobic. Otherwise, that center of the membrane is going to repel them, right? So lipids can do this. CO2 gas can do this. Oxygen gas can do this. Nitrogen gas can do this. And I'm going to say some water. I know water is polar, but the thinking is that because water is so teeny, it really is just an oxygen with two protons, those two hydrogens, that it can actually manage to slip through. So like over kind of here towards the right, see how we have some unsaturated fatty acids? We're thinking that those are probably the easiest region for water molecules to be able to scoot through because there's a little bit more space between those fatty acid tails. So some water can move through. That is kind of an exception to your non-polar hydrophobic rule here. Right. And again, we do just want to take note that we are moving from a high to a low concentration. So I'll say high to low, that should be an arrow, concentration. I'll abbreviate concentration there. And again, we are going from high to low simply due to kinetic energy. So I'll say this is due to kinetic energy. And we are going directly through the phospholipids, directly through the phospholipids themselves, right? So let's go ahead and add some of those to this drawing. Usually, oxygen is going to be in high concentration outside the cell, and the cell needs to import that. So we're going to have a high concentration of oxygen outside, diffusing along its concentration gradient. Again, the concentration gradient just means going from a high to a lower concentration into the cell. For CO2, it's usually the opposite. For CO2, because your mitochondria are producing CO2, usually that's more of like a waste product that's leaving the cell. So a high CO2 concentration inside to a lower CO2 concentration outside. Water can go either way depending on how much water that cell needs. Usually it's actually exiting the cell. And we talked about lipids also, so we can add testosterone here. or estrogen. Those can also fit right through the phospholipids simply based on passive transport. No ATP required, and they can move right through the phospholipid bilayer. All right, that's the simplest. It's obviously the least expensive. We're going to come down here and look at what happens when these molecules aren't hydrophobic, what happens when they're polar, what happens when they have a charge, and what happens when they're hydrophilic. So, oops, down here towards the bottom, I have facilitated diffusion. So facilitated just means helped. So these polar molecules, these hydrophilic molecules, are going to need some assistance, and proteins are going to help them move across the membrane. Because this is diffusion, we are still going from a high to a low concentration. So we're still going from a high to low concentration, but we're going to do this through a protein channel. That is the key difference here. That is the key difference. So let's go ahead and scroll back up. And the reason why we need these protein channels is because if you are hydrophilic or polar, and you try to go through the phospholipids, you're going to get blocked. You're going to get rebelled against. You're going to get rejected because of the hydrophobic center of this membrane. So you need a passageway to make it through, and that's why you have these protein channels. So one protein channel that we've talked about before is GLUT. Remember, that's a glucose transporter protein. Usually cells are going to want to be bringing this in. So there we go. There's our hexagon glucose molecule, our monosaccharide. Again, glucose is polar, so it does need a transport protein, and that can diffuse right on through this channel. Remember, no ATP needed. This is simply passive diffusion based on a high concentration of glucose hitting other glucoses and other molecules and passively diffusing to a lower concentration of glucose on the inside of the cell, right? Water is a similar story. So water, again, it can go directly through the phospholipids like we just talked about, but usually you're going to have what are called aquaporins. Aquaporin is a type of passive transport protein that is unique to water, so we can add that. Other materials that might need a transport protein include your body salts, so sodium ions, chloride ions. Again, those are flat out charged. They're not going to be able to go through the fatty acid tails in the lipid bilayer. They're going to have a protein to help them out. And again, this is just simple diffusion still through a protein channel, so it's facilitated technically, but this is just kinetic energy moving these things. There's no ATP required here. So let's say that one is a sodium channel, maybe this one right here is a chloride ion channel. And again, these protein channels are specific. It's not any willy-nilly thing can fit through them. It is specific to that molecule. Other things that might need to go through are amino acids. AAs, amino acids. Every amino acid has its own transport protein, has its own protein channel here. It's not a one-size-fits-all. Not every single amino acid can make its way through the same channel. So there are 20 amino acids. There are 20 different channels, and that is an important point. So I'll go ahead and type that here. Different for each amino acid. Remember, these channels and these proteins are specific to the materials that can move through them. And one more that we have that I'll talk about is ammonia, NH3. It's a waste product from cell metabolism. That's going to want to leave the cell because it's going to be produced in the cell and have a high concentration inside, and it's going to want to diffuse down to a low concentration outside the cell. So all of these things are still moving without energy from a high to a low concentration gradient, but because they are polar or charged or hydrophilic, they do need to move through that membrane with the help of a channel protein. All right, let's move over to active transport. So I'm going to grab the hand, slide back over here to the top. Active transport requires ATP. You require ATP, and again, that's an energy molecule. Another way to state this is that you are going against the concentration gradient. Against the concentration gradient, which means you are moving from a low to a high concentration. Right? It's kind of like stuffing a suitcase. You want to forcibly pack molecules together in a really, really high concentration because naturally they're going to want to go from a high to a low concentration. So that takes energy to reverse that from happening. Just like when you pack a suitcase, you got to take a lot of energy to stuff all those clothes in. It's the same here. You need a lot of energy to stuff all those molecules onto one side of the membrane because naturally they don't really want to do it. So one example that we're going to look at here is the sodium-potassium pump. Every time you see the word pump, you need to know that that is active transport. You're actively going to pump something through, which means you are going to require ATP. You are going to require ATP. So in this sodium-potassium pump, the cell is going to naturally have lower levels of sodium inside the cell, and it's going to want to pump them out of the cell. So sodium is going to come out of the cell from a low to a high concentration. So we had 2 sodiums here. I'll double that, we'll make it 4. And the sodium-potassium pump can handle 3 sodiums at a time. So see these 3 little kind of notches here? 1 sodium can fit in each of them, so actually 3 sodiums are going to make their way out for every kind of rotation of this pump. And at the same time, potassium is going to come in. So outside of the cell, potassium is usually high. Potassium is usually low. So I'll have 2 triangles. And we're going to pump them, again, from low to high, just like stuffing a suitcase. We're going to cram all of these potassiums in here against their will using ATP. And the sodium-potassium pump can handle 2 of those for every 3 sodium. So see, there's only 2 notches here. So 2 can fit in this pump at a time. And we're going to pack them over here on the inside of this membrane. So high potassium, right? And that's how a sodium-potassium pump works. So 3 sodiums get pumped out from a low to a high concentration, and 2 sodiums get pumped into the cell from a low to a high concentration. Again, this is active transport, and it requires ATP, and a giveaway to that if in a diagram is if you see ATP or also if you see the word pump, you know it must be active transport. Another type of active transport is for protons. So scroll down here. I've got a proton pump right here. Again, you do need ATP for this. So I'll put a little star there just to highlight it so you have an ATP molecule. Typically cells are going to want to pump protons out, and I'll show you why here in a second. So inside, we'll put maybe 3 protons. Remember, proton is just a hydrogen. It's just a positive charge. So we have a low amount of protons inside the cell. You are actively pumping them to the outside of the cell, so we'll draw a higher concentration out here. So the protons move from a low to a high concentration gradient via a pump that is specific to protons with the help of ATP, because this is active transport. So we are going against the gradient. Against the gradient. Whenever we say we're going against the gradients, we're saying we're going from a low to a high concentration because we're stuffing the suitcase. In this case, we're stuffing it full of protons. All right, so when you talk about proton pumps, you can obviously therefore talk about pH. So when cells pump protons out of the cell into the extracellular matrix or whatever else is out here, you are increasing the concentration of protons, right? Which means you are increasing, well, I don't want to say the same thing twice. I meant to say decreasing. You are decreasing the pH. right? You are making the surroundings more acidic. When you increase the concentration of hydrogen, you decrease the pH of, in this case, the outside of the cell. All right, let's scoot down here. There is another proton pump right here because I want to show you how this can work with other pumps. So here's a proton pump again. And we're going to return to this in just a second, but I want you to look at this co-transport. Sometimes cells can move multiple things at once. So we're actually going to show you how cells can move sucrose. So sucrose is usually going to be outside the cell. If you're a plant, you want to bring this in. So this is why plants is out here. Plants are going to do this much more than animal cells are. Sucrose is going to make its way into its little kind of notch here in this co-transport protein, but it itself won't be able to move through. It actually will need some help in this case, and that's why we have this proton here, right? The proton is actually going to move simply based on diffusion. So the proton, oops, there's no S there. This is not fascist. This is facilitated. Facilitated diffusion. So, and we have high concentration of protons because of this proton pump. So these two pumps actually kind of work together. So because you're pumping protons outside of the cell, this high concentration of protons due to this proton pump can then turn back around and move from a high to low concentration just by facilitated diffusion, so no energy needed, thanks to having this proton pump here. When these protons do that, glucose takes advantage of it. Glucose is going to piggyback and take this opportunity to get carried along with the proton and move into the cell. Right, so here you actually have a coupling. There's a coupling of proton pumps. They worked of membrane pumps, of protein pumps. They work together. The proton pump pumps hydrogens to the outside of the cell, and because they are now in a high concentration outside the cell, they can diffuse with the help of this transport protein. So this is facilitated transport. They can diffuse back in to a lower concentration and carry along this sucrose with them. So it's like a win-win situation here. The cell has to pump protons just to bring them back in and piggyback sucrose along with it. So this is called co-transport. If you drink things like Gatorade, not only do they have sugars, but Gatorade also has body salts. They also have sodium, they also have ions, they also have hydrogens in them. And that's because the company knows that in order to uptake these sugars, these disaccharides that are bigger than just a monosaccharide like sucrose, they're going to need to provide the co-transport proteins with those ions and with those solutes. So that's why things like Gatorade will not only have sugars, but they'll also have body salts and protons in them as well, because the co-transport proteins need them to get the sucrose into your body cells. All right, the last little bit here, the last little bit down here is, this is if molecules are just simply way too big. You have to flat out engulf them or spit them out. So this is a membrane. I'm going to zoom in over here. It's only aligned as a membrane, so I know we've done like the double membrane or the double structure over here with the bilayer, but here I'm just simplifying it so it's just a single line. These, I know they're little small dots, but these are supposed to be very large molecules. Monosaccharides are gonna just diffuse through, like we talked about before, but potentially some disaccharides, polysaccharides, and anything else is gonna need to be kind of engulfed. So these molecules are going to come into the cell, and the cell membrane is going to make a pocket. It's going to pinch around that molecule and just kind of cut it off. So eventually this pocket will eventually close and pinch off, and that vesicle carrying those proteins or carrying whatever that is. It could even be a virus or it could be a prokaryotic cell. Moves into the cell to get digested and broke it down. So these vesicles will actually become vacuoles. They will mature to become vacuoles. And you can do this vice versa too. You can have a vacuole full of waste that's too big to pass through the cell membrane otherwise. So these little vacuoles will fuse with the cell membrane and release the contents. So if you're taking materials in, that's called endocytosis. And if you're pushing materials out, that's called exocytosis. And that is for bulk transport of massive molecules. So I'm going to go ahead and write that here. Let's say that this is for bulk transport in and out of cell. In, out of cell. Of large molecules. Of large molecules. That's the key thing. All right, so let's go ahead and talk about, I'm going to move on to diffusion of water and osmosis. So we're over here in this middle third. Excuse me for the coughs and the sneezes. I have a little bit of fall allergies. All right, when water is diffusing, we don't just say the diffusion of water. We give it a special name, and that name is osmosis. Right? Water is so important, it gets its own name. Water is diffusing in this case still, so we are going from a high to a low concentration. I'm going to abbreviate high and low as H and L. And again, this is diffusion, so there is no ATP required. This is spontaneous due to the kinetic energy of these molecules bouncing off of each other and dispersing as they do so, right? So I'm going to label this cell B, and we're going to label its environments A. So A, the environment, in this case, it could be a beaker, or it could be your bloodstream. Maybe this is a blood cell that's kind of getting carried around in your bloodstream. But any environment your cell finds it's in, it could be A in this case. We're just kind of doing a demo here. All of these little dots are some kind of solute. They could be glucoses, they could be potassium, they could be sodium, they could be amino acids, but it's some kind of solute that is in A but not in B, right, in this case. So I'm going to show you the same thing over here. So here's the same thing. So here's side A with all the solute in it, and here is side B. So these two are equivalent. This drawing just has the cell, and this side just has kind of another side of this box. And they're separated by a semi-permeable membrane. Semi-permeable, that means water can go through, but solutes cannot. Solutes are too big, and we can manufacture these membranes in a lab. But your naturally occurring cell membrane is also semi-permeable in the same way. So water can move through this membrane, but the solutes cannot. The solutes cannot, right? For kind of a helpful hint, anytime you have a semi-permeable membrane, I want you to assume that the solutes are not going to be able to go through unless they're hydrophobic. So I'm going to say, unless told differently, biological, so biomembranes, do not let solutes through unless they are hydrophobic. So for simplicity, you can always go ahead and make that assumption when we talk about semi-permeable membranes, right? So let's look at what's going to happen, right? What's going to happen in this case when we have a semi-permeable membrane with more solute in A than we have in B, right? So this is side A, and here is side B, right? And again, the dots are solutes, and this dashed line is a semi-permeable membrane. Let's say that this is actually, let's just say this is glucose. Let's say this is glucose. So this membrane is not permeable to glucose. What's going to happen to the movement of water here, right? Well, side A has a higher solute concentration. Just look how much more glucose there is on side A. Look how many more dots. We call that side hypertonic. Hyper means you have a lot of something. Like when you're hyper, you have a lot of energy and you're all over the place, right? The side with less glucose is called the hypotonic side. So the side with less solute, in this case, the solute is glucose. Hyper and hypotonics are always relative. If you didn't have side B here, there wouldn't be anything to compare side A to. So you have to have a comparison. Side A is hypertonic compared to side B. Side B is hypotonic compared to side A. And again, you're always looking at the solute concentration. That's key. Always the solute concentration. Another way to say that is side A has less water, right? Because everything else in here is water, right? So if you look at, let's look at side B first. Look at all this extra space. There's not a lot of solute, so look at all of this water, right? So when you're hypotonic and have low solute, you also have high water concentration. And when you are hypertonic and you have a lot of solute, obviously that solute takes up space, which is space water can't inhabit, so you're going to have less water. It's an arrow. It's an up arrow for high water, a down arrow for low water. You're going to have low water concentration. Remember, the brackets, just like in chemistry last year or 2 years ago, means concentration. Well, we know that water is going to diffuse across the semi-permeable membrane. It's going to go from a high to a low concentration. So water is going to go from side B to side A. And that's just simple diffusion of water. That's just osmosis, right? Also, let's make sure to put this in brackets. It's low water concentration and high water concentration. It's not just low water and high water. So don't forget the brackets. All right, so we answered which way does water move. It moves from high to low concentration. What happens, therefore, to side A? Well, side A is going to get bigger. It's going to swell. If side A was a cell, it could potentially burst. Right? And side B, because it loses water, is going to shrivel. Or get smaller. All right. So down here towards the end of this third, there are some synonym vocabulary words that I want you guys to be aware of. Anytime you see low water potential, that is the same as saying you have low water concentration. If you say you have high molarity, what you're saying is you have a high solute concentration, just like we had in side A, which is the same as saying you have low water concentration. So saying that these two things, saying you have low water potential and saying you have high molarity, they're synonyms. They mean the same thing. If you ever see that you have the word osmolarity, I just want you to cross off the word or the prefix os. It's just kind of distracting. It really just means the same thing. So high molarity means the same thing as high osmolarity. And it also means that you have high osmotic pressure. Osmotic pressure is the pressure needed to stop water coming in. So if we look back at A, because water is going to diffuse from a high to a low concentration, water really, really wants to move to side A. So it's going to take a lot of pressure to counteract that and prevent that from happening. So when you have a hypertonic solution, you're going to have low concentration of water, and a synonym for that, so another way to say low concentration of water is to say that you have low water potential, you have high molarity, you have high osmolarity, and you have high osmotic pressure. So similarly, on side B, it's the flip. It's the inverse. If you have low solute concentration, you're hypotonic, that means you're going to have high water concentration, which means you're going to have high water potential. I'll abbreviate potential. High water potential. It means you're going to have low molarity. You're going to have a low concentration of solutes. It means you're going to have low osmolarity. And it means you're going to have low osmotic pressure. Low osmotic pressure. All right, last little bit here. Let's scoot on over to this last third. We just talked about water potential, so let's go ahead and do it in a little bit more detail. This is a Greek symbol that means water potential, so I do want you to recognize that. The equation for water potential takes into account two different types. One is for the solute, that's what the S is for, and one is for the pressure. So I'm going to show you how solute and pressure affect your water potential. So let's look at solute first. Let's look at solute potential first. Here's our semi-permeable membrane. Side A has more solute, side B has less solute. So without doing anything here, the default, the baseline, is for water to move from a high to a low concentration. I'll put the brackets there, that just means concentration. So in this case, water is going to move from side B to side A. That is the default, right? I'm going to change colors here. Oops. I don't know. Let's just say this is a different color. If we increase the solute inside B, so I'm going to add solute. So I'm adding all of this solute, lots of solute everywhere. Now side B is super salty. We've increased the solute inside B. Now water is going to want to move from A to B. Right, so in the baseline case, we went, I guess this should have really said high, just to keep things consistent. It went from high, that's a mess, I'm sorry. From high on side B to side A, from high water concentration to low water concentration. So that arrow should point to side A. And then when we increase the solute in side B, now all of a sudden we have higher water concentration on side A. We have lower water concentration on side B. So now water is going to move from side A to side B. Remember, the solutes do not move, but the water does. So what we can do, and this is the whole point, is we can move water by changing the solute concentration. So by manipulating solute concentration, organisms can change water movement. Organisms can change water movements. If they want to bring water into the cell, they'll pump ions and solutes into the cell to draw water into the cell. If they want water outside of the cell, they'll pump ions and solutes outside of the cell, and water will follow. It's always going to want to move from a high water to a low water concentration, and you can achieve that simply by manipulating solutes. Another way to achieve this is to apply pressure. So here we have the same thing. Again, water is going to move down its concentration gradient from a high water concentration on side B to a lower water concentration on side A because of all this solute. But if we apply pressure to side A, we can reverse that, right? So if you apply pressure, let's say we have a little piston here, we force this down, we can actually push water from side A to side B if you apply pressure to this side. So we can force and we can manipulate the movement of water by applying pressure. So here's an application in real life. Reverse osmosis does this, pretty much with the same diagram. If you have salty water, like in the Middle East, in the United Arab Emirates and Kuwait and all those areas that have very, very low natural water sources, they have to get a lot of their water from salt water, they will do reverse osmosis. So they will have setups much similar, very similar to this piston setup, osmosis. and they will apply pressure to the salty water. They will force fresh water, in this case to side B, and that they can drink that, and that's potable fresh water. There's two downsides to this. It takes a lot of energy to push that piston. Remember, this piston does not want to get pushed down. You have to apply quite a bit of force to get that piston to go down because this is so highly concentrated with salts. So you do need a lot of energy for this, and it is pretty energy inefficient. So it's not an ideal process to use. But it also creates brine. So if you are in AP Environmental Science, brine is this very, very salty solution that's left over, and there's really no good solution to disposing of it. You can't really dump it in the ocean because it'll kill the sea life. It makes the water super salty, and it'll kill it. You can bury it, but then you can't plant crops there or use that land for anything productive because the salt draws water up from your plants, and then the plants shrivel up and they die. So brine is usually something that you want to try to avoid, but that it is an application process in real life. I'll do one more real life application process with Twinkies. So the cake in a Twinkie, so your Twinkie has that kind of spongy cake on top. It has that spongy cake on the bottom. And it has this filling in the middle. So it's going to look like a hot dog, but it's a Twinkie. So this is some kind of creamy filling. And the cake is pretty dry. You don't get a Twinkie and the Twinkie is all soggy and sopping full of water. It's a dry cake, which means it has low water potential, right? So the cake on either side has a low water potential. Well, how do they do that? How do they make sure the cake has low water potential? Because this cream is nice and moist. It's not wet, it's not liquid, but it is really nice and moist. So the cream has high water potential. Remember, go back over here if you need to refresh on what that means, right? High water potential was side B for us, right? So high water potential is going to want to give water to the low water potential side. So these Twinkie cakes on the top and bottom are going to want to collect water. Water's going to want to move to the cake side from the cream. So how does it not do that? Because your Twinkie cakes are not soaking wet, right? So what they do is they manipulate the solute potential, right? What do they do to that cream to help the cream retain water? They pump it full of sugar, right? This cream is super, super, super sweet, right? They add all of these sugar solutes, glucoses and whatever, to the cream. That's going to help prevent water from going from a high to a low water concentration, and it's going to retain water because that cream is so hypertonic. Right? So that's how, even though your cream has high water potential, your cakes stay nice and dry. This is the same thing. Oops, let me just go. So what they do is they increase the solute potential. So the subscript here is S for solute. And they do that via added sugar. This is the same thing for pastries and pies and any kind of cake that you might buy. For Doritos when you're, not Doritos, for burritos when you go to Chipotle or whatever, the filling that they put in, the meat filling or the whatever you want in your burrito is usually very salty. That's to help increase the solute potential of the filling and to decrease the amount of water that seeps out into your burrito or that seeps out into your pie crust, right? So it's no accident that your pies or your burritos that you buy are so either sugary or salty. It's to help prevent the cake or the burrito itself from getting all wet. All right, one thing I do want you to pay attention to when you go over this, especially on the movement of water, is that this is not something you're just going to be able to sit back and memorize. You're going to have to be able to apply this. You're going to have to be able to identify which side is hypertonic, which side is hypotonic, which way water is going to move. And you're going to have to be very comfortable with all of these synonyms, the low water potential, the molarity, the osmolarity, the osmotic pressure. The more versed you are in this, and again, this is all below low water potential or low water concentration, because these are all synonyms for low water concentration, right? These on the right are all synonyms for high water concentration. So practice with this, right? Practice with it, get comfortable with it, because we are going to start to apply this and see this routinely throughout the year. So I hope this helped, and I will see you guys next time.